using the adapter hypothesis, first theorized by Crick, that explains how information encoded in DNA is used to specify the amino acid sequence of proteins. This image right here, simplified manner, first with transcription, which is the movement of DNA in forming RNA. More specifically, for this example, mRNA, which is messenger RNA that acts as the messenger between DNA and protein production, and tRNA, which is transfer RNA that brings amino acids to mRNA to create the growing peptide chain that will become a protein. This occurs in ribosomes that read the mRNA to form proteins. This is known as translation. tRNA in a two-dimensional image would look like this T shape here, with the anticodon at the bottom and the amino acid at the top. But for simplification, I'm using this box method here, which has the anticodon at the bottom and the amino acid at the top. The ribosome, which is this big object here, reads or translates the mRNA. When it reads this from left to right, you can see with the 1, 2, 3 reading, the transfer RNA comes in with the anticodon, links to the matching sequence at either the A, P, and E site. First the A site, then the P, then the E. You can see here, if this was to link to this one, at the A site, the amino acid at the top, then links to the amino acid in the P site, and then the transfer RNA moves to the E site that is then ejected from the cell. This continues and is translated further into a polypeptide chain occurs, and you can see here that eventually it will be ejected as a newly formed protein with specialized function. Now you can see why he would call this the adapter, because these, almost in a modern day sense, look as plugs that perfectly match the sequence within the ribosome. And all it would take would be for these to mismatch a single, maybe even letter, to 3 sequences of a codon, and this would completely change the sequence of the protein that would be formed in the end. So this is a very highly complex and very organized function of tRNA, or transfer RNA, that functions using the anticodon to match with the mRNA, to use the codon of the amino acid at the top to match with the one beside it, and then expel from the ribosome as a newly formed protein. This could result in misfunctions, could cause cancer or malfunctioning proteins, to DNA that could result in the death of the cell or organism.